Hey everybody, and welcome to the 16th ibiTea. We are very excited to have you and glad that so many wanted to participate today. So my name is Louise and I will be the moderator. Today we are very lucky to have Dr. Bill Cheng from the National Chongqing University in Taiwan. He will introduce his latest publication, which illustrates a successful model of how immune cell trafficking can contribute to efficient transport of drugs in the body during chronic disease. And Dr. Cheng will do his talk the next 20 to 30 minutes, and hereafter we will have approximately 10 minutes for any questions that might come up. So I hope everybody has a tea or coffee with them. At least I have mine here. So I would say let's get started. And Bill, you are free to share your screen.
Okay. Can you see the slide?
I see the slide
Okay.
and also the presenter mode. Looks
good. Okay, I'll start right away. So, good morning to those in Europe and those to US, and good afternoon to those in Asia Pacific. So, my name is Bill, and I'm from the Graduate Institute of Biomedical Engineering in National Chongqing University in Taiwan. So, it's a very honor to be able to give this talk. So, today, as Louis has pointed out, today the focus of my talk will mainly coming from a paper that we recently published in Journal of Control Release. But the main focus will be on the in vitro assay where we have used some of the slides that developed by EBDs to demonstrate how our drug delivery system was able to penetrate the tumor microenvironment. So just start with some background. So for those who work on drug delivery, we notice that getting a drug to a target cell or a target microenvironment is a very challenging task. So this is a very recent review paper published by Warren Cheng's group. And in this review article, you basically point out if we take tumor or cancer example, for every 10,000 for every 1 million drugs you administer to the patients, probably only 7,000 reach to the tumor microenvironment. So in other words, the delivery efficiency is only about 0.7%. And if you look at how many actually reach to the tumor cells, and the numbers roughly 14, and if the drug target is somewhere in the nucleus, probably only 1. So as you can see, this delivering issue is a big problem right now in the medical world. So it's not because the drug do not work, but rather it never reached to the target site. And if you think about it, this is not a problem. It's not just a problem for nanoparticles or any other form of drug carriers. This is also same thing also apply with antibody drug conjugates, which we often call it the magic bullets. So this is a very recent paper just published in 2024 in Cancer Discovery, which pointed out that in terms of the delivery efficiency in terms of for antibody drug conjugates, the delivery efficiency is only less than 1%. In other words, more than 99% end up in other parts of the body. Same thing with exosome. This is another excellent paper published in 2015, which pointed out that all the exosome that you administer into the mouse model, very, very few, if you can see through by the IVUS imaging, very few actually reach the tumor. In fact, majority of them get stuck in the lung, liver, spleen, or other organs. So you can see the drug delivery is a massive problem now in our today's society. So why this is a big problem? Why are we having this issue right now? For those who work on drug delivery, we notice for over the past, I guess, 3 to 4 decades, we really rely on what we call the EPR effect. So this which EPR stands for enhanced permeability and retention effect for drug delivery. In other words, as a tumor, so we take cancer example, as a tumor starts to develop, the blood vessel around in the tumor microenvironment will become leaky, either due to tumor angiogenesis or because the tumor was secreting enzyme undergo tissue remodeling. So we somehow hoping that the drug that we injected into the patient will magically end up in the tumor microenvironment because of its leakiness. However, it has been coming where as pointed by this review paper that I've put on the slide here, that the EPR effect only offer probably less than twofold increase in terms of the nanodrug delivery compared to a healthy organ. So in other words, that's not a lot, all right? So it's that's not good enough. And if you look at in some other tumor, let's take breast tumor or pancreatic tumor, for example, there's no really EPR affected, especially with pancreatic tumor. And so those tumors are often classified as cold tumors because the blood vessel, just the number of vasculature that's happening in those areas is just very low. So it is very challenging now is in how can we actually get a drug into the tumor microenvironment, and how can we do it without relying on the EPR effect. So that is a challenge that we, I think everyone have to address. So now this doesn't happen with a single cancer. It happens with a lot of cancer, even with colon cancer, which is highly vascularized, like of the lung, the brain, and it doesn't really matter which cancer type you're talking about. The delivery efficiency is all less than 1%. So this is a big problem that I guess pharmaceutical companies as well for us that works in academia all have to address. So what my group has been working on is, or trying to come up with an alternative way in delivering drugs, is to utilize this immune cell called monocytes. So for those who know immunology will know that monocyte is a part of the innate system. And when you have an abnormal tissue develop in your body, so it doesn't matter whether infection or whether the tumor, it's basically any abnormality happens somewhere in your body, there will be some sort of activation of the monocyte recruitment, and the monocyte will come out from the spleen or bone marrow, as pointed by this picture here. And these monocytes almost like have their own GPS. They know where the abnormal tissues are. So this is just say if I have an injury in my heart, like a myocardial infarction, for example, the monocyte will know where they need to go to the heart, and they will travel directly go to the infarct area and not to the healthy tissue. Same thing with tumor. If you fortunately have a tumor develop somewhere in your body, the monocyte will know where the tumors are located, and they will, once they come out from the spleen or the bone marrow, they quickly will travel towards the tumor site, and they will, in fact, they will go to the tumor site directly without stopping at the healthy tissue at all. So when we saw that, we thought, this might be not bad, I guess, a carrier for drug delivery. So our strategy is very simple. So let's just say if a patient today unfortunately has a tumor develop, monocyte recruitment will happen. We then would develop this drug carrier that will able to hitchhike onto circulating monocyte. So in this case, we don't extract the monocyte out from the patient or from the animal model. Instead, we inject the drug carriers directly into the circulation, and that these drug carriers will able to hitchhike. So they carry peptide that recognize a specific receptor that you found on monocyte, and that so that the drug carrier will able to hitchhike onto the monocyte. However, they will remain anchored on the surface, so they will not get internalized. This is very important because if they do get internalized by the circulating monocyte, you get premature drug release, and that's not what we wanted. So we want the drug carriers to remain on the surface of the monocyte after they're able to hitchhike onto the cells. The monocyte then able to carry the drug carriers into the tumor microenvironment, and once they have done that, the monocyte will become macrophages. The macrophages then will internalize the drug carriers that were originally anchoring on the cell surface, break them down, and then you have drug release. So this is a simple cartoon diagram of our drug carriers. It's basically you have this liposome as a core. Now you can switch this to nanoparticle LNP, lipid nanoparticles if you want to. You can So you basically can just adjust the formulation very easily. But for this particular study, we use liposome because we're encapsulating doxorubicin. And on the surface of the drug carrier, you have PEG and peptide that basically was recognized specifically for a receptor that you found on monocytes. So this is the first assay we have done. So basically, we create a library of these monocyte mediated drug carriers. That's what we call them, MMDCs. And so to see which one works well, we did a lot of screening. And this is one of the assays that we have done. So we basically exposed different drug carriers or different combination of drug carriers that we came up with to the THP1, which is a monocytic cell line. And this is a suspension cell line. And once we incubate the monocyte or THP1 with the drug carriers, we left them in the incubator for about 4 hours or so, and we take them out and do some imaging. And you can see even after 4 hours of exposure, and if you use confocal microscope to scan along the z-axis, you can see that majority of the drug carrier, which shown in red, in fact remain anchoring on the surface of the cell. And to demonstrate this interaction very specific for monocyte only, we did a flow cytometry analysis. And you can see that you see a lot of interaction with THP1, whereas they show no interaction with the HUVEC. Now, this is very important because you can imagine if the drug carrier have any interaction with the endothelial cell, the drug carrier may induce undesirable thrombotic events in the animals or in the human blood vessel. The next thing we want to show that this interaction is a cell state dependent. What I mean by that is that the drug carriers will remain anchoring on the cell surface when the cells are in a monocyte state. However, if we differentiate them into macrophages, the localization of the drug carrier will change, as in the macrophages will uptake them. So we just done a very simple assay here. So first we expose the monocyte, which is a suspension cell line, with the drug carriers. So the monocyte is shown in green and the drug carriers is in red. You can see that the drug carrier remain on the surface of the cells. And when we do some chemical treatment to activate those monocytes to become macrophages, immediately you can see all the drug carriers being internalized by the monocyte-derived macrophages. So this shows that our drug carriers, in fact, is a some sort of cell state dependent function. As in, depending on the state of the cells, its localization will change. Okay, so I think onto the most interesting part. So the stuff I just showed you earlier in the previous slides was static, and obviously monocyte in our circulation is a dynamic environment, and it's very difficult to, I guess, visualize them in an animal model because you can't really mount. You can, but it's very difficult to mount the mice onto a confocal microscope, for example, because it's very challenging. Plus, you need some assistance from people that have experience working animal. So it's a really challenging task. So I decided to look for a simple option, and this is where I came across with one of the ibTea's products, which is the use like one lure. So when I see this, I thought, oh, this is great. Perhaps we can see how we can use this slide to study how our drug carriers were able to interact with the circulating monocytes. So we use a 0.8 channel lure, and it's a glass slide. And these are the results. So we first inject the monocyte into the slide and let it circulate for a while at 37 degrees. Then we inject the drug carriers into the slide and let them circulate for about 4 hours together. After 4 hours, we then mount the slide onto a confocal microscope, and we continue to let the perfusion pump running, and we start to take images. So this is a snapshot. Later, in the next slide, I'll show you the video, but right now I'll just show you the snapshot of the results. So you can see that with our drug carriers, they were able to anchor on the surface of the circulating monocyte even after 4 hours of circulation together. Whereas with the control will use the classical formulation of pegylated liposome, which is a formulation that's commonly used in the clinic right now. And you can see that with the pegylated liposome, it shows no interaction with the circulating monocyte. And if you take them out from the channel slide and do a flow cytometry analysis, you can see that the number of drug carriers able to anchor or interact with the THP1, the number is extremely high. It's almost rough, it's more than 40%, whereas with the control, it's very low. So now I'm just going to play the video to show you the results. So the one up here is the control group. And the one down the bottom here is our drug carrier MMDC. You can see this runs very fast. So I think we're producing greater than 4 dyne per centimeter cube, if my memory serves me correctly. You can see that the one with our drug carriers, you can see, you probably can see that, okay, you will see that, but it's red particle anchoring on the surface of the green colored THP1. Okay. All right. So that was the hitchhiking experiment. So the next experiment we want to do is that can the monocyte able to carry the drugs and penetrate the endothelial barrier into the tumor microenvironment? And to do this, we use the IBITEA's use like 1 Lewis 3D, and we reckon, and so far we had a lot of success with this slide. It is probably one of the best slides we ever bought from IBITEA. It's great, we really love it. And what we did was we established 2 different microenvironments. The first one is a very simple study where we simply just have a collagen gel inside the space of the I guess the gap within the lure. And once we fill that with the collagen gel, the next thing we did was, we then we put a layer of endothelial cell on top of the gel, so forming an endothelial barrier. So we let it perfuse for a while. Then we inject the monocytes, then followed by the drug carriers. So if you look at figure A is where we have not inflamed the endothelial layer at all. You can see that very few endothelial cells have attached to the very few of the THP1 have attached to the cubic forming endothelial layer. But when you if you pre-treat the endothelial barrier with TNF alpha, so you inflame them, and followed by monocyte, and then by the drug carrier, you can see that a lot of the monocyte would attach to the inflamed endothelial layer. And if you focus on any of those cells, you can see our drug carriers remain anchoring on the surface of those monocytes. All right, and if you measure the cytokine that was secreted in the media, you can see that those treated with TNF alpha will secrete a lot of NCP1, which is an important cytokine that involved in attracting the monocyte towards the inflamed endothelial cell. And if you count the number of the endothelials attached to the inflamed number of THP1 cells attached to the inflamed endothelial cell, you can see that the number is much more greater to those that were pre-treated with TNF alpha compared to the non-treated. But I think the most important data is the figure shown by figure E here. If you do a scanning along the Z-axis, you can see that the monocyte that were attached to the endothelial barrier were able to penetrate or infiltrate, the trans-endothelial migrate through the endothelial barrier into the gel underneath. And if you blow up, so you basically amplify one of the cells, you can see that our drug carrier will probably, I think by this stage, it will already be uptaken by the cells because they become macrophages already. All right, so this is just a video show you the interaction between the monocyte to the uninflamed endothelial cell from the model I showed earlier. So this is the inflamed one. You can see that the TH1 have no interaction with the endothelial layer at all. It just runs through it. And let me play the this is the inflamed one. So you can see already there are some cells attached to the endothelial layer already. If you have a look at this one, this is undergo what we know called tethering. So it's kind of like loosely, or they try to grab on the endothelial, the inflamed endothelium, and you can see it already stop. You can see that it's already stopped, and it's probably now ready to undergo transmigration. Now, unfortunately, I couldn't capture a real live image of the transmigration process because I think it will take. I just don't have enough. Don't have the equipment to do that, but it would be great if we can able to capture the transmigration, like a live image of the transmigration process. Yeah, unfortunately, we couldn't do it here. Okay, so this is then so that was with a TNF alpha treated model. So by now, some of you probably thinking, all right, what happens if you have a real cancer cell in the gel? Would that able to inflame the endothelium? After all, that's more closer to the real pathological scenario. And the answer is yes. So here we have again using a 3D Lua slide. What we did was within the gel we have the human breast cancer cell MDA MB231, and we let the cancer cell sit in the gel for about one overnight. So next day we coat the collagen gel with the endothelial cell again, forming an endothelial layer, and we perfuse about 2 days, roughly about 2 days. Then on the third day or the fourth day, we inject the monocyte and then circulate for a while, then inject the drug carriers. So within 4 hours, you'll see that a lot of the THP1 will attach to the endothelial barrier, okay, and that they will actually undergo transmigration as well, just like shown in the previous slide. So the blue color here represents the cancer cell, and the green represents the transmigrated THP1. And again, if you blow one of the cell up, you can see that our drug carriers remain probably already uptaken by the cell by this stage since they're about to differentiate into macrophages. So I think this is probably the most exciting data from this paper. This shows that we're able to use the EBD system to monitor the whole process. In fact, when I submit this paper, the reviewers really like this system. One of them say they're gonna contact EBD. I don't know whether he or she has done that. They were very interested and to see they were very excited to see you can actually use a very simple assay like this to study how drug delivery system interacting with all these cells in a very simple, I guess, in a very neat in vitro assay. Okay, and so, but this is just a control group. If you repeat this experiment with HEC293T, which is a non-cancer cell line, you don't see any inflammation happening at all. In fact, very few monocytes attached to the endothelial barrier, and you don't see any transmigration at all. Okay, so this what we saw in the previous slide is very specific, cancer specific. Obviously, to demonstrate the in vitro the EBD in vitro assay is reliable, we also did animal studies. So this is just animal data to show you the data we got here really match what we saw in the in vitro assay earlier. So this is a hitchhiking efficiency data where we inject the nanoparticles into the mouse model, then we extract the monocyte out with the mice. We extract the monocyte out with the drug carrier, and the flow cytometry analysis to demonstrate the hitchhiking efficiency is roughly about 40% to 50%. So match perfectly to the EBD to the assay that we did with the EBD slide. And this is the biodistribution assay to show out our drug carriers were able to enter the tumor microenvironment at least tenfold higher compared to the pegylated liposome, mainly through the monocyte hitchhiking. This is an assay where we simply use the drug carrier to encapsulate doxorubicin, and we inject it into the mice. And you can see that the mice that receive our drug carrier with the doxorubicin have low expression of Ki67 in the tumor tissue. lower expression of incoherent, but high expression of activated caspase-3. So this is the last slide just to summarize our finding. Basically, here we have demonstrate that you can use the EBD channel slide to answer a lot of questions. In this case, we demonstrate that our drug carriers can interact with the monocyte, and we can actually capture the live imaging of that in a dynamic manner, and it has a very high targeting specificity for tumor tissue and not for healthy tissue. So this is my last slide. So with that, first I just thank the students from my lab that are involved in these studies. Also had to thank Cheng Li from Academia Sinica, who's a co-corresponding author on this paper. Cheng and his students are the one that are basically responsible for the manufacturing of the nanoparticle and the drug carriers. Also like to thank Dr. Yao Cheng from the Zhonghua Christian Hospital. He's I guess he's the only oncologist in this study who has been providing a lot of suggestion how to interpret the data. Also, I thank Dr. Miran Zhu from the Institute of Molecular and Translational Therapeutic Strategy at Hanover Medical School in Germany. She has been providing a lot of excellent suggestion on how we should go about with studies. And we have to thank the funding agency, this is the National Science and Technology Council in Taiwan. And lastly, I had to thank IBD, especially people like Tina, Louise, and Bettina have been hassling a lot with some of the technical problems. And also, I have to thank Blossom Technologies, which is the supplier in Taiwan. Thank you. I'm more than happy to take any question.
Thank you so much. That was great. And I just wanted to remind people that it's not possible to raise the hand and speak, but you can use the Q&A section and include your question in there, and then I will cover them one at a time. But I would say let's get started with the questions if you're ready, Bill.
Yeah.
Perfect. Okay. So were there any technical challenges when encounter when establishing this 3D model in the Microslide 1 Lure 3D?
That's a very good question. Yes, we have. I think for those who work with EBD channel slide, I think one of the challenges we always face is bubble.
Yeah.
Yeah. So the way we do it or try to eliminate the chance, the possibility of bubble formation is before we connect the perfusion pump to the slide, we let the perfusion pump, so with the media in the syringe, let them circulate themselves.
Mm-hmm.
So we place the pump into the we place the microfluidic system into the incubator and let the media to circulate within the syringe for a while. And then we connect that to the channel slide. We find that if you do that, you actually eliminate a lot of chances where you have bubble formation. The other thing is that when we fill the channel slide with media, we did it with syringe, so syringe needles. We have tried use normal pipette tip before, but I don't know why. I'm not sure because of pipette or whatever, but we usually get a lot of bubble formation as well. But with syringe needles, we don't get that. So that's why I had to buy a lot of syringe needles for this kind of experiment.
But yeah,
so we
that's
that
good tips.
Yeah. So I noticed that when we use syringe needles to inject media into the slide, you get less chance of bubble formation. Yeah.
Yeah, at least also with the incubator, it makes sense because when you then
Yeah.
heat it up, gases are also released from the material and media. So that's always a tip that we also give.
Good. Yeah.
Okay, more questions are coming in. Is it possible to count the transmigrated cells that transmigrate through the endothelial layer? For example, to extract them from the slide and count them with flow cytometry?
That's an excellent question because a reviewer asked me that question before. The answer is yes. We did not do it for this paper, but we tried before. It actually works. You can. But I think it would be great if your cells are able to express some fluorescent protein beforehand. It allows you to separate them out from the rest with other cell type that you have in the channel slide. Yeah, but the answer is yes.
Yeah, perfect. Another one came in, so thank you for excellent talk. I was wondering whether you could develop a similar study for circulating CTCs. Also, which velocity in the MF chip did you find to be optimal for this experiment?
Oh, with CDC. Um, we actually, I think just a few weeks ago, we did an experiment where we just have these. So we let the MDA cell. So just let me recap what I just presented earlier. In this paper, the cancer cell were just we just mixed cancer with the gel. So the cell were in single cell type, in a single cell form. Obviously, as time goes on, they will start to aggregate together, form this mini-like spheroid. Now, if you begin your experiment with a cancer spheroid in the gel, what you will find is that they will undergo metastasis. And what we discovered that if you have endothelial cell layer forming on top of the spheroid, you will see cancer cell coming out from the endothelial layer into the liquid layer. So where you had where the channel where you had the media. And if you repeat this experiment with the 3D Lewis slide, you know how you have a three like space or three gap, you will see that the cancer cell migrating out from spheroid will jump to the next space, the next little gap, and forming a new spheroid themselves. So the answer is yes, you can. I guess I'm not sure would you call that CTC or not because it's a cell line. So, but you can use that to study metastasis, and the experiment actually repeatable. We actually tried that a few times, but it's just that we haven't published yet. Yeah,
That's really cool. I never thought about that application in this slide.
yeah.
And also there was like a sub question regarding the optimal, how do you say, speed of
Oh,
the circulation. So did you use
oh,
specific flow rate? Do you remember?
we actually tried a lot of fluoride. I think all of them is all fine. We didn't see any difference in terms of monocyte hitchhiking. We don't see any differences. Obviously, when you do a slower shear rate, the interaction might happen much more quicker.
Hmm.
But at the faster rate, they still happen. It just takes more time.
Yeah, but that makes sense as well. Yeah,
Yeah,
maybe you need to try a bit to find the
yeah,
optimal one.
yeah.
Then there is this continuous question regarding this count of the transmigrated cells. So how exactly do you extract them from the slide? And maybe I can answer a bit here. So at least the EBD slide is made with a thin polymer on top at the bottom. So in principle, you can remove this and get access to your samples within the well of this microslide 1 Lure 3D. It would also be possible to extract them like you do in any other um, labware format, so trypsinize it or use acrytase compare. It is, however, how to say, it is, I'm missing a word. It is depending on how what you have in your wells and what you want to extract from the wells.
Yeah.
But I don't know, Bill, did you do it in a specific way?
Yeah. I use collagenase to
Hmm.
degrade the
That's
collagen
also
gel
possible,
to
yeah.
liberate the cell out. So that's why in the earlier I mentioned whether your salesperson tag will be important at this stage because you might want to separate them out. So for our study, we had the spheroid in express red fluorescent protein and the monocyte express green. I'm trying to get someone to help me to get the endothelial cell to express blue fluorescent protein. Yeah.
Okay. I hope that answered the question.
Yeah.
At the moment, there are no further questions. There's just a comment regarding the opportunity to maybe connect with you, but I will reach out later regarding that.
Oh yeah, that's fine.
Um, then maybe a question here. So, why was the microslide 1 Dur 0.8 chosen for the hitchhiking experiment? So, why
Okay.
this exact channel
Yeah.
slide?
I think this is a very important question for those want to work with circulating cell. We try I think we tried 0.4 before, but what we notice in when the I guess when the depth of the slide is too low, circulating cells tend to form aggregation during circulation. So they kind of like run each other and bump each other, and they kind of block the entire circulating they basically block the entire circulation. So you got this clot thing happening. So we know that 0.8 is the best. It's highly repeatable.
Yeah,
Yeah.
so there's room for the cells to, yeah,
Yeah,
flow
exactly,
by.
exactly,
Makes sense.
exactly. because even if I try a lower cell, like for example, if I try with 1,000 monocyte and I still use 0.4 chance, like you still get this aggregation happening. Yeah. But where
Okay.
with 0.8, if you put even with plus 1 million cell, they still flow very fine. You can see, lovely cell just circulating, in the slide, without any aggregation at all. Yeah.
Satisfying.
Yeah.
Okay, I do not see any further questions, so I hope everybody got a chance to ask. And otherwise, if questions comes up later, you're thinking about a specific setup or something, then always feel free to reach out to us. Then I think it's just, how to say, left to thank you again for your time and your great talk, and then have a great the rest of the day as well.
Yeah. Thank you. Thank you, everyone.
Perfect. Then I will end this EBT and hope to see you at a future one. Bye
Yeah.
bye.
Thank you. Bye bye.